Principal of Art, Commerce and Science College, Chhattisgarh. Dr. Jaipurkar sir has joined with us through the online mode. So please give a loud round of applause for them. I welcome the chief guest of today's function, Honorable Dr. S.G. Thakur sir, HOD of Chemistry in RDIK College, Bhandara. I welcome Honorable Professor Rathur ma'am, HOD of Chemistry in Vidyabharati Mahavidyalaya, Amravati. I welcome the convener of today's program, respected Dr. Gafari sir, and I also welcome all the guests and the resource person of today's workshop. We have the privilege that persons having a spectacular personality are our today's guests. Hence, I feel it my honor to invite the chairperson of today's program, Honorable Dr. Vijay Tompe sir, to occupy their respective seats. Thank you, sir. I request chief guest of today's function, Honorable Dr. Thakur sir, to occupy his respective seat. Thank you, sir. I request Honorable Professor Rathur ma'am to occupy her respective seat. Thank you, ma'am. I request the respected Dr. Ramteke sir, Principal of GS Tompe Art, Commerce and Science College, to occupy his respective seat. Thank you, sir. I request respected Dr. Gafari sir to occupy his respective seat. Thank you, sir. I request respected Dr. Chaudhary sir to occupy his respective seat. Thank you, sir. I request respected Professor Dr. Masad sir and Dr. Rahul sir to occupy their respective seats. Thank you, sir. Today, technology plays a great part in our daily life. These technological advancements are present in our daily routine. Technology plays an incredible significant role in improving access to education, and the education sector is one of the fields that benefits from services of computational tools. And hence, when it concerned with the chemistry, computational tools plays a very important role. And hence, undoubtedly, chemistry is super important in developing and advancing various aspects of life. Chemistry comprises of variety of branches and sub-branches. Computational tools in chemistry comes under the branch called computational chemistry. It is quite innovative branch of chemistry which uses computer programs to solve complex chemical problems. Hence, the main motive to carry out the workshop is to learn about the tools that help individuals learning and also to learn about the latest computational tools in chemistry in developing of productive skills. And the topic to be covered in this workshop is fundamentals of computational chemistry, basics of aid drugs design. So I hope this workshop will be beneficial for you. Deep is a symbol of knowledge. The light of this lamp has power to dispel the deep darkness. Let us pray to Saraswati Mata that this lamp of knowledge continue to shine in our college. Nahi dhyanena sadrusham pavitram hi vidyate. Hence, I would like to request all the dignitaries on dais to candle the lamp. you, everyone. It is our good fortune to welcome our today's guest, which possess innumerable virtues that hypnotize others towards them and make their place in the heart of others. Just as flower, however small, attract others with its fragrance. आज इथल्या थोरवीला चंदनाचा गंध आला, महाविद्यालयाचा परिसर पाऊलांनी धन्य झाला. महाविद्यालयाचा परिसर पाऊलांनी धन्य झाला. It is the moment of extreme pleasure to welcome the guests present here to attend this workshop. So first of all, I would like to request IPSC coordinator and the convener of this workshop, Dr. Gavade sir, to welcome the chairperson of today's program, Honorable Dr. Vijay Tompe sir, by offering a bouquet. I would like thank you, sir. I would like to request Dr. Chaudhary sir to welcome the chief guest of today's program, Dr. Thakur sir, by offering a bouquet. Thank you, sir. I would like to request chief guest of today. I would like to request Dr. Nidhi Dixit ma'am to welcome the chief guest of today's program, Honorable Professor Rathod ma'am, by offering a bouquet. I would like to request Professor Yavle sir to welcome respected Dr. Ramteki sir by offering a bouquet. I would like to request Dr. Ubrate sir to welcome the Professor Dr. Masud sir by offering a bouquet. Thank you, sir. I would like to request Mr. Yogesh sir to welcome Dr. Savarkar sir by offering a bouquet. Thank you, sir. I would like to request Ms. Bhakre ma'am to welcome respected Dr. Gavade sir by offering a bouquet. Thank you, ma'am. I would like to request Ms. Tetu ma'am to welcome Dr. Chaudhary sir by offering a bouquet. Thank you, ma'am. Now, I would like to invite Dr. Nidhi Dixit ma'am for the introduction of today's guest. Now, I would like to request Honorable Dr. Thakur sir to inaugurate today's workshop.
Now, symposium workshop concerning inaugurated.
Thank you, sir. Now, I would like to request Dr. Nidhi Dixit ma'am for the introduction of today's guest.
Thank you, Pranali. Friends, because of lack of time, we have to start our first session. And in this session, we have our main resource person is Dr. Abhinav Kumar. Before starting this introduction of Dr. Abhinav Kumar sir, I humbly request Dr. Hemant Chandak sir to accept the honorable chair of the session. Thank you, sir. Now it's my pleasure to introduce you all with our honorable resource person, Dr. Abhinav Kumar sir. Dr. Abhinav Kumar sir has completed his graduation and post-graduation at HCPG, followed by completing his PhD from Department of Chemistry, Banaras Hindu University in 2009. Currently, he is working as professor at Department of Chemistry, University of Lucknow. While telling about his academic details, I would like to mention that he has published near about 190 research papers in various international journals, and also published 208 books. He has delivered very impactful lectures as an invited talks in various conference, seminars, and symposiums. His teaching areas involve specialization in spectroscopy, organometallic chemistry, inorganic chemistry, and multinuclear NMR. He has a huge experience of research in various areas such as coordination and organometallic chemistry, source precursors for binary and ternary chalcogenides, metal organic frameworks, coordination polymers, and dye-sensitized solar cells. So, ladies and gentlemen, please give a big round of applause for such an eminent person is present here. Now,
I many thanks, but I request you to kindly allow me to speak.
I request Dr. Subbarhande sir to welcome Honorable Dr. Hemant Chandak sir by offering a bouquet.
आप जो है उनको बोल दीजिए ना कि मेरे को अलाउ करें स्क्रीन शेयरिंग करने के लिए. हाँ हाँ, एक्चुअली होस्ट डिसेबल्ड पार्टिसिपेंट्स स्क्रीन शेयरिंग.
Without wasting much time, I kindly request Honorable Dr. Abhinav Kumar sir to share his knowledge with us.
Please give me some time. Actually, host will be making me co-host, then I can share my screen. Many thanks, and I have now right now having very less time to display what we are doing with computation. So I would be quickly introducing some basic aspects associated with basics of computation. So could I start? Can I start? Could I start? So I'm starting. So today I would be trying to focus on some basic concepts associated with computation, and I would be starting with a very general definition associated with computational chemistry, which was taken from Wikipedia, and it indicates that this is a branch of chemistry that utilizes computers to assist in solving some chemistry related problem, and it always uses the results of theoretical chemistry incorporated into efficient computer program to compute or calculate the structures and properties of molecules and solids. It can, in some cases, definitely would be producing or would be engendering some results which are unobserved during certain chemical reaction, and that is why this type of or this branch of chemistry is now primarily deployed in designing of new drugs and materials. And definitely, if we wish to design new drugs or materials, we must have certain types of models. So we move towards the molecular modeling phenomena, and this particular molecular modeling always apply theoretical approaches and computational technique to model different types of structures. And not only they model the structures, rather they also predict certain properties associated with the compounds and materials. And using this, they are used for studying different types of molecular systems. It may be a very small 2-atom molecules and maybe moving up to crores of or hundreds of atoms or thousands of atoms. It may be simple organic compound or any inorganic coordination complexes, or even in polymeric system also. So what is the major goal of computational chemistry? It is to create efficient mathematical approximation. Definitely, without mathematical approximation, one cannot execute any computational chemistry using computer program. And they used to calculate the properties of molecule and to apply these programs to concrete physico-chemical systems. And what basically we do with the computational chemistry, use geometrical structure, and using those geometrical structure, we can have certain insight regarding the ground and excited state of different phases of matters like gas, liquid and solid. And not only these geometrical parameters, rather we can also assess the energies or geometrical parameters associated with conformation and tautomers. They also provide us energies. And whenever we talk about weak interactions, it may be hydrogen bonding, unconventional hydrogen bonding, van der Waal interaction. They also engender the energy parameter, which we call as interaction energy. They also provide us with electronic charge distribution, dipoles, and higher multiple moments also, which is helpful in predicting certain very important. There is some sort of disturbance, I think. So any sort of important material property, which we call as non-linear optical property. And whenever we talk about vibrational spectroscopy, that is frequencies as well as intensities, UV visible spectroscopy, electron spin resonance, NMR spectroscopy, and apart from that, molecular orbital energies and shapes using UPS and X-ray systems. Sometimes called it as computational spectroscopy. So why we compute any molecule? Because we can have certain insight regarding the molecular structure, molecular properties, and spectroscopic observable, using which we can challenge, not only challenge, even help the certain scientists or researchers or investigators in the experimental area. I request everyone to mute themselves so that I could say my words. They can also helpful in obtaining some unmeasurable properties like spin density. Remember, there is only one neutron diffraction analysis with the help of which we can calculate the electron density, or X-ray spectroscopy, we can calculate the electron density, but spin density could not be calculated or computed with the help of any experimental technique. So we used to perform computation to obtain insight regarding the spin density of any molecular system. Apart from that, we can also do this very tedious or typical calculation, which is known as benchmarks for parameterization. So these are very important names one must keep in mind whenever he or she is performing computational computations for their molecular system, and they must remember these several names. I'm not going to name this because we are already running away from time, but I would be sharing this slide, and they must remember these names. You all know the Hitler, and he was the person who proposed a very important technique interactions. Then London Kohn-Sham orbital is very important. Then Poppel, who proposed certain electron density computation or calculations. Then Leonard Jones potential, a very important potential, who developed the linear combination of atomic orbital methods. Slater, Mulliken, Pauling, Boyce, and Gauss. So all these workers have contributed significantly in the development of computational chemistry. So whenever we talk about any molecular model, we always talk about the structure, and that particular structure have to be correlated with its property. So this structure property relationship could be executed in two different ways. And first is the experimental data. If it is existing, then that could be correlated with the structure. But if it is the experimental data is not existing, we can compute different things, and that particular computation can produce certain calculated data, which is always possible. So one can amalgamate this experimental data and calculated data to produce a correlation, and with the help of which we can propose different types of phenomena. So from here, we can say that the computational chemistry is one of the most rapidly advancing and exciting field in natural science today, because many experimental chemists, in fact, I am basically the experimental chemist, uses different kinds of spectroscopy in the research, although we are not spectroscopists. Likewise, we are also experimental chemists. We are applying computational techniques or tools as another weapon in our arsenal to propose different parameters. Yes, Nandu? Actually, my network is very perfect. My network is perfect. Actually, I'm using around 500 Mbps speed. Okay. So what shall we learn behind the to have certain insight regarding the computational chemistry, so we must learn the theory behind the molecular modeling. To perform such particular molecular modeling, we must have certain molecular visualization package. I hope that in proceeding sessions you would be having certain molecular visualization package, and you would be going through those packages to have insight regarding the molecules. To use program package designed for molecular electronic structure theory, and to execute calculations at different levels of theory. It may be ab initio calculation or density theory, density functional theory calculation, and other calculations also, to do different calculations at different levels of theory and to interpret the results, to make correlation between the experimental and theoretical data. So to execute these targets, we are having certain different components such as Hartree-Fock theory, density functional theory. We use basis sets, electron correlation model, and this basis set superposition error. This is a very important technique or keyword which we basically employ whenever we use to address or we attempt to address the weak interactions between two or more than two different molecular systems. And this could be achieved only with the help of geometry optimization, which is correlated with vibrational spectroscopy, because if we perform geometrical optimization without executing vibrational spectroscopy, that particular optimized geometry might be a local minimum, not a global minimum. And then apart from that, we can compute NMR spectra and ESR spectra of the desired compound. And apart from that, we can address the electronic structure using UV visible spectroscopy. So can we do research using pure theoretical studies or not? So definitely we can publish our data by executing only and only theoretical studies, but coupled experimental and theoretical investigations on the structure and property of any type of molecular systems are gaining attention nowadays, and they used to get published in such type of journals. And I also had published in some Royal Society, in American Chemical Society, Wiley, Taylor and Francis, and Springer, etc. So I'm rushing towards the methods. So there are different methods using which one can perform computation, and out of those methods, the most rudimentary method I used to recall is the empirical approach. And out of this empirical approach, the first method which I would be discussing here is the molecular mechanics method, which is sometimes regarded as empirical force field calculation. So remember, this particular computational technique or computation is based on classical mechanical model of molecular structure. And here what we do, that we used to consider any atom in that particular molecule as a single particle. So that particular single particle is simulated in that molecule, and the potential energy of the molecule is calculated, which is based on a given force field. So here, remember, the electrons are not considered explicitly, rather they find their optimum distribution once the position of nuclei are known, and that is always correlated with this very celebrated approximation, that is Born-Oppenheimer approximation. So remember, in this particular computational technique, atoms are considered as a sphere with a given radius, that is van der Waals radius, and polarizability as well as net charge. So you all know van der Waals radius, this may be the expression. Therefore, we are having some polarizability associated with this particular any molecular system form electric susceptibility, and that particular polarizability can be computed with this expression, and van der Waals radii could be computed easily with second expression. So here, interactions operating between the atomic species or single particles are based on classical potential, and that determine the spatial or space distribution of atoms as well as their energies. So the potential energy of molecular system here is always equal to the summation of covalent energy and non-covalent energy. And I had attempted to generate a diagram associated with carbon-carbon system where carbon can form either single bond, double bond, or triple bond. So you all can observe that they are almost having a similar sort of minima, but their bond distances or bond energies are different. Apart from that, with the help of this calculation, one can have insight regarding the dihedral angle. So for dihedral angle, we can have different patterns. So if dihedral angle are ranging between 0 to 360 degree, we can have different parameterization associated with potential energy parameter. So here the covalent interaction is always related with the bond energy, energy related to angle, energy related to dihedral angle, while non-covalent interaction, which is a non-bonded interaction, is always correlated to electrostatic as well as van der Waals interactions. So each molecular mechanical model is characterized by particular force field. This is very important, and it is equal to interatomic potential functional form and parameter set. And definitely it is used to describe the potential energy of a molecular system. I attempted to add as many text as possible so that everyone can have certain weak insight regarding the computation that can have a feel. So it is derived from experiment and high level quantum mechanical calculation, and it is always based on a set of equation. Remember, it is always based on the set of equation that defines how the potential energy of molecule varies with the position of its constituent atom. A series of atom times is also important, which defines the characteristic of an element with a specific chemical context. It may be any chemical context. So the atom type, remember, depends on hybridization, charge, and types of other atoms to which it is bonded. Another parameter set that fits the equation and atom types to experimental data. So parameter set contains force constant. So force constant, suppose you all know that carbon-carbon single bond, double bond, and triple bond definitely would be having different force constant, and structural data, that is bond length, valence, and dihedral angle. So all atom force field are there, and united atom force field are also existing there, and all atom force field contains parameter for each atom in the system, while united atom force field computational technique treats carbon and hydrogen atom in suppose molecular system CH2 and CH3 groups as a single interaction center. So there are certain disadvantages associated with molecular mechanical method, that is each force field although provided good results, but it is limited to a very small class of molecule for which it had been parameterized. So no force field is in general is generated. Since empirical methods do not consider electrons, they cannot describe the formation of bond or cleavage of bond. And many molecular properties which depends on very small change in electronic effects are not always reproducible with the help of molecular mechanical method. So molecular mechanical methods are generally used for energy minimization, and this is useful for energy barriers between different conformers, steepness of potential energy surface around a local minimum, protein folding kinetics, protonation equilibria, active site coordination, designing binding site. And with the help of this, the results of molecular methods can lead to the optimized geometry, can produce potential energy surfaces, and docking, which is gaining a significant amount of interest nowadays. Docking, I do not do this area. Then vibrational spectroscopy, and also some thermochemical parameters. Next calculation or computational method is molecular dynamics method. So it follows the temporal evolution of systems of interacting atoms. So by integrating their equations of motion, that is force is equal to mass into acceleration, it is being given a set of position and velocity. Evolution in time for this particular type of computation is completely determined, and it is a statistical mechanical method that provides configuration distributed according to some statistical distribution function. It is having certain mathematical concepts, so I'm trying to avoid this, but it is based on the force acting on atoms derived from this particular formula. And the simplest choice for this particular parameter V is to write it as sum of pairwise interaction operating between either two atoms or more than two atoms. So the simplest choice is the pair potential, in which the total potential energy can be calculated from the sum of energy contributions between pairs of atoms. Apart from that, it is based on Leonard-Jones potential, which is having this particular expression, having sigma and r, and intramolecular bonding interactions here are also have to be considered, and those interactions would be leading to some stretching, bending, and torsion energy parameters. So molecular dynamics method are also offering certain advantages, such as it could be utilized to treat a very large molecular systems, thousands of atoms, and that is why it is used in biophysics to address the nature of proteins and viruses, as well as in material science. And it is having also some disadvantages, that is, it is not able to model bond formation breaking, since electronic structure methods does not enter into this models. Now, the second method is the quantum mechanical approach, and this quantum mechanical approach is having one very important computational method, which is regarded as semi-empirical method. It is having variety of computational technique which could be regarded as AM1, PM3, triple P, Endo, Mendo, Zendo, etc. And it is an approximate method based on Hartree-Fock formalism, in which some quantities such as electron repulsion integrals are taken from experiments. But in this computational method, some small quantities are usually neglected. Some quantities are estimated by fitting to the experimental data, and the empirical parameters and functions are used to compensate for errors introduced by neglecting some integrals. They model, remember, only valence electron or pi electrons, as in Huckel's method. The core electron in this particular semi-empirical methods are treated together with nuclei as one effective core potential. And this is energy which is equal to this expression where Z prime are reduced nuclear charges. And this technique could be executed for those molecular systems which comprises of hundreds of atoms, and can be used to investigate the nature of ground as well as excited molecular state. So they are parameterized to reproduce experimental reference data. Just one minute, please. Experimental reference data or accurate high-level theoretical predictions, as they contain set of parameters. The total energy in this particular computational technique is represented as the sum of electronic energy, net negative, and the core repulsion, which is net positive. So a training set of molecule in this particular technique is selected, chosen to cover as many type of bonding situations are possible, and it leads to a non-linear least square optimization procedure with values of variable adjustable parameters as variable, and set of measured properties of the training sets as constant to be reproduced. So the measured properties which can be obtained from this technique are heat of formation, certain geometrical parameters, that is bond parameters, bond angle, dihedral angle, torsion angle, etc., dipole moments, and ionization potential. So depending upon the choice of training set, the exact number of types of adjustable parameters and mode of fitting to experimental properties, different semi-empirical methods have been developed and incorporated into some semi-empirical software. This is the link for MOBAC, and here are certain computational methods in this semi-empirical process. So if we move from semi-empirical process to non-empirical process, what we see that non-empirical process do not require empirical parameter, and hence they can be used for any type of molecular system, but they are limited to tens of atom, where the molecule is having around 50-60 atoms. They can be used to study ground and excited molecular states. They are reliable for strong as well as weak, that is non-covalent interaction. And amongst these, the first method is the ab initio method, which is based on wave function, and second is the density functional theory method, which is based on electron density. So based on they are based on the following principles, that in non-empirical methods, nuclei and electrons are distinguished from each other. Electron-electron and electron-nuclear interactions are considered explicitly. Here, interactions are governed by nuclear and electron charges. So potential energies, not only potential energy parameter, rather also on the electron motions. Interaction determines the spatial distribution of nuclei and electrons as well as their energies. So what level of theory we use to execute? So level of theory is always related to remember two very important aspects. First is method and second one is basis set. So it is determined by the property being calculated and the cost of calculation in terms of computer usage. So suppose we are having a very small computing system, suppose a PC equipped with 16 GB or 8 GB RAM, we cannot execute a very big computation. So cost of calculation is always a very important aspect. Second, in general, the higher level of theory that requires greater or large computational cost. So for example, Hartree-Fock based ab initio methods are more costly than semi-empirical methods. Correlated methods, depending on sophistication, add a significant additional cost to Hartree-Fock method. So for the Hartree-Fock and density functional methods, an additional factor is the quality, that is the size of basis set which we are deploying. So in general, the larger the number of functions used in basis set, the more flexible or better would be the basis set. Each additional function, remember, adds extra cost to computation. So I attempted to compute this particular molecule, and for this computation, I had to spare around 121 days, 18 hours, and this much minutes and times to finalize my calculation. Likewise, I attempted to perform some cumulines interactions, pi-pi interaction, again for different types of computational system experiments. From AM1, it took only 6 seconds. For density functional theory, it required around 1 hour, but for molar placid computation, it required around 8 hours to execute or to finalize the calculation. So it is the front view and the view of the molecular system. So in summary, we can say that methods of computational chemistry may be empirical, and in it we are having molecular mechanics and molecular dynamics. In turn, quantum chemical or quantum mechanical methods may be semi-empirical or non-empirical in nature. And since today is the resurgence in non-empirical method, we are having ab initio method and density functional theory method. And ab initio method is based on wave function, while density functional theory is based on electron density. So using this non-empirical method, we can execute different types of calculations such as single point calculations. It produces energy, wave function, and other requested properties at single fixed geometry, usually done first at the beginning of the study. I used to perform any computation in this manner, beginning of the study, on a new molecule to check out the nature of the wave function. Apart from that, it is also frequently carried out after a geometry optimization with a larger basis set or more superior method than is possible with the basis set and method used to optimize geometry. So definitely geometry optimization is very time taking. So I used to execute computation for big molecular system by employing small basis set, and after getting the small basis set, what we see that we get a reasonable molecular geometry, 3D geometry, and using that for optimized coordinate, we perform higher level calculations. So these calculations are performed to obtain basic information about a given molecule, check a molecular geometry to be used as a starting geometry in an optimization process, compute more accurate energies and other properties for an optimized geometry at a lower theoretical level. So its one cycle could be represented as f phi equal to epsilon phi in metric form. It is written as Fc is equal to Ce, which produces a first self-consistent field cycle. So second is the geometry optimization calculation. So it calculates the wave function and energies at a starting geometry, and then searching for the geometry which will be giving the lowest energy, that is local or global minimum energy. It is used to locate the lowest energy geometry or structure of a molecule or a molecular complex. It is used to locate the transition structure on the potential energy surface. And I attempted to represent the two cycles of self-consistent field, and it is represented like this. So here we can obtain several parameters like local minimum, the lowest energy point in a limited region of potential energy surface. Second, most important is the global minimum, the lowest energy point anywhere on the potential energy surface, and a saddle point also, maximum in one direction and minimum in all other direction, and correspond to the transition state structure. So all minima plus saddle point always lead to the formation of a stationary point where F might be 0 and energy summation would be equal to 0. So geometry optimization usually locate the stationary point closest to the starting geometry. Apart from that, we can execute the conformational search, which is necessary to distinguish between a local minimum and global minimum, and how it is executed by altering the initial geometry slightly, usually by altering the dihedral angle and then performing another optimization. Additionally, we can execute some frequency calculation to identify the nature of a stationary point on potential energy surface. It checks if an optimized geometry corresponds or not to a minimum energy or not. To predict the IR and Raman spectra of a molecule, it may engender frequencies, intensities, and normal mode, and in turn it could be correlated with the energy parameters, thermodynamic energy parameters. To compute force, our force constant for geometry optimization, to compute 0-point vibrational energy, thermal energy corrections, enthalpy and entropy, that is what I was talking, to compute polarizability and hyperpolarizability tensors, but should only be carried out at the geometry. Remember, the last point is very important. It should be carried out only when the geometry obtained from an optimization run and the same basis set and method have to be utilized. So for a local or global minimum, all the calculated frequency will be positive. But for any stationary point other than minimum, some of the frequency may be negative, which is alternatively termed as imaginary frequency. Apart from that, we can compute magnetic properties also. They are having certain types of error like NMR spectrum of any compound, which might be giving chemical shifts and spin-spin coupling, and ESR spectra. But for this ESR spectroscopy, which produces hyperfine coupling constants and hyperfine coupling tensors, there is one very important program named as ORCA, ORCA, developed by Frank Nies from Bonn, Germany. That gives a very reliable result. So these are the properties which is usually calculated not only by Gaussian 9, rather by some other advanced versions. And these are certain keywords using which we can calculate those molecular properties. And then this is a very simplified geometry which few days back I had computed for you, which is showing how this molecule is transforming to a global minimum. So I had to stop these work, in fact, and I am now focusing on metal organic framework. So I intended to show what basically we are doing. We are using some metal organic framework as a sensor for nitroaromatic sensing, and this was the paper I had mentioned only review papers, luminescence sensing of nitroaromatic compounds which are explosives by crystalline porous material. So we had computed we had attempted to address the enhancement or quenching in emission through computational technique, and recent development associated with coordination polymers and metal organic framework, and how they are used as luminescent material for plethora of application that had already also been reviewed. Apart from that, I am working also in the area of metal organic frameworks, catalyzed photo degradation of organic dyes. And since I already conveyed that I'm basically an experimental chemist, so we used to synthesize variety of compounds also. So we are focusing on multivariate isoreticular synthesis also. So we are using a technique, reticulate chemistry or reticulate synthesis, using which we are producing variety of metal organic framework. So last year or in 2020, before COVID, we had shifted our focus from material aspects to biological aspects. And here you can see here that we had used metal organic framework as a nano platform to generate reactive oxygen species for cancer treatment using chemodynamic therapy. And this is relatively a recent work where biomimetic cell membrane tumor therapy application have been executed. So my special thank to, I have another assignment also. Special thanks to Dr. Nand Kishore Gawhale, and special thanks to my mentor, Professor Rajen Prasad, who was my senior at Banaras Hindu University, who taught me how to compute. So this is my department at Banaras Hindu University, and this red region is my lab at the basement, where I had started doing research, synthesizing different compounds, computing their applications. And this is the area, this is my University of Lucknow, where I am starting and had developed many different aspects associated with chemistry. So thank you, and I request everyone to keep computing rationally for different types of molecules. Thank you. So if everyone allows, should I take leave because I have another assignment within 10 minutes?
Sir, Abhinav Kumar sir, am I audible?
Yes, I can.
For enlightening us with the basics of, I think
I am leaving now because I'm already late by 15 minutes. Sorry.
we can go ahead. Thank you. Thank you on the behalf of the organizing committee. Thank you as the chairman of this session, Professor Abhinav Kumar. You can leave. Thank you. So for rest of the participants, I will just conclude what has been presented by Professor Abhinav Kumar. In his session, he has tried to cover so many things, and especially how the computational chemistry has arrived. This was the focus. Mostly, he has focused on the theoretical part. Theoretical part. All the basics of the computational chemistry he has covered, and he especially demonstrated how we can compute property and we can supplement our research. Whatever the statistical chemistry we are doing in our lab, it can be supplemented well by computing the properties. Many things he has covered, the theory behind the molecular bonding, then different methods, especially molecular mechanics, and then the quantum mechanical method. In the molecular mechanics, he narrated he narrated in addition to the molecular mechanics, he also taught the basics of the quantum mechanical calculation, in which he talked about the Hartree formulation, non-empirical methods like ab initio calculation, DFT calculation. I would like to share with the participants that I also had done some DFT calculation, and we did some synthetic part in our lab, and we did some computational calculation using DFT. And because of the DFT, we could publish our paper in a good journal. So supplementing our research with the computational calculation will help us in going ahead with our research intentions. I had a few questions, but probably these people will address it in the afternoon session. You want to add something?
So I have a question as you gave us about the DFT. How we can implement that DFT in this company?
My experience, what I did in my case, actually, what we did, we were trying a multi-component reaction in which we were trying to synthesize highly functional elements by pairing. And what we did a DFT calculation using basically 26 given GD basis set, and we have optimized the transition state for the plausible mechanism. 3-4 plausible mechanism we have written down on the paper, and we have optimized those transition state using DFT. And we propose that on the basis of the DFT calculation, we propose that how the catalyst is actually playing a role in the reaction. So that part we have made that we So that was the story. Moreover, I would like to again share one more thing that using the process 09, our master students and undergraduate students they have taken some research projects. And in that they have done some simple calculations. One of the practical that we have conducted in our MSc curriculum presently, MSc partner, where we have each confirmation of weakness. Like we talk about anti-confirmation, each confirmation, but students are not able to know what is the actual quantitative confidence there. So you can just optimize the staggered and eclipse confirmation of the eclipse, compare the energies, and you fact that it will be around 3 km. So that way student can get insight. So probably I expect you people to please address that so that whenever we go back to our call those practical. And I would also request you people to if you can tell us is there any Android version? Because providing computer to the 20 students, 25 students in the college is sometimes not possible. So if we can do certain calculations using mobile application, probably we can give them a training and we can ask them to conduct the calculation at home and come back with the results. Some of the Excel sheet and those sheets can be discussed in the lab session probably. So these are some of the things which we expect from you. I think that's all because we have so many things to cover in our session ahead. So thank you everyone and thank you organizer for giving me an opportunity to chair the session.
Welcomes you all to the second session. Welcome, Dr. Chandak sir, for offering. Okay, thank you, sir. Now, this session, the chairperson has already taken his seat. Today's second session resource person is Dr. Vaishali Patel, ma'am. She joined us by online mode. Here, I'm to introduce her. Dr. Vaishali ma'am is an associate professor in Department of Pharmaceutical Chemistry, KIT Group Office Officer, Delhi NCR, Ghaziabad since 2017. She completed her pharmacy degree in 2000 and post-graduation degree in 2007. She did her PhD in 2014 from Birla Institute of Technology, Germany, SRA, India. Now, about her experience. She has experience in application of computer-aided drug design, QSAR, and related medicinal chemistry approaches for biological important classes of organic compounds. She also has experience in handling complete computational software and analytical data interpretation, that is IMS. So, now about the research. Madam has published more than 60 papers in national and international journals. She has written 60, more than 16 chapters in 16 books, including elsewhere Springer Nova publications. She received funding from various national and international funding agencies like SCRT, ESP, DAV, DVD, ICMR, AKTU, RSP. Adam's publications on designing of drug on Alzheimer's disease and COVID-19 are notable here. Also, I am so excited to hear her at this moment. I welcome Dr. Vaishalya Patil ma'am to deliver her lecture. Please, ma'am.
Thank you so much, sir. Am I audible to you? Hello? Am I audible to you, sir?
Yes, ma'am, you are audible.
Hello?
Yes, ma'am.
Okay.
The recording has stopped.
Am I audible
Yes, ma'am.
clearly? Shall I start?
This meeting is being recorded.
मैं धर्मेंद्र sir, can you hear me?
Recording has stopped.
अच्छा अच्छा आता ऑडिबल आहे सर? So shall I start then? If there will be any disturbances from my end, kindly let me know. Either you can call me or you can put in the chat box, because I am not able to listen anything from your end. So sorry for that. First, I will like to congratulate all the organizers of this national symposium, that is on sophisticated chemistry techniques for computational tools and chemistry, computational tools which are used in chemistry. So, first of all, this is a very interesting topic, and I am really happy that I got opportunity to be part of this symposium. And first of all, I will like to congratulate the convener of the symposium, as well as Vijay sir, as well as other organizing committee. And that there are so many participants and very very labs basically, whether it is India or and thank you so much for this opportunity. So. For this today's session, I have taken a very simple topic like I'm not much aware that what kind of participants are there, but there might be people who are having basic knowledge of computational chemistry, as well as there might be persons who will be using it for the first time. So I have tried to convey the concept, or to cater the both the areas. First of all, I will be giving an overview of why we need the computational drug discovery, or what is computer aided drug design, and how it is going to be helpful us in regularizing or making our drug discovery approach verified one. Yes.
Vaishali ma'am, am I audible? Hello.
So that has approaches which can be assessed to everybody, take up some steps where the mankind is needing novel drug for curing the infections with it, and time was lacking. So in those cases, we were reliable. Excuse me, there is call from sir. Yes, I think it is visible now. So during that pandemic time, we were covered in a shorter period of time. And for such kind of cases, drug discovery process to be very fast, very rational, where like for any drug moiety to be used in clinical patients, it has to be very safe, it should be potent, and in addition, sorry, there is a railway line behind my chamber, so I will not be able to hear you properly. there is a disturbance for a second. So please bear with that. The qualities of a drug that is required when it's going to be used. It has to be safe, it has to be potent, there should be no side effects, toxicity should be taken care. Uh,
Madam, can you hold for 2 minutes, please?
Hello, हाँ sir, हाँ जी.
Here, I have some technical problem.
अच्छा अच्छा, मी तर फुल स्क्रीन केलेलं आहे
The
सर.
recording has stopped.
एक मिनिट, मी परत रीशेअर करते आता.
Madam, 1 minute, please hold on.
हाँ हाँ सर.
We have some technical.
ओके ओके सर.
सर झाले कशासाठी डाउनलोड तुमचे माझ्या एनर्जी सूट मध्ये थोडासा प्रॉब्लेम समजून हे एनर्जी होतं का स्केल चालू आहे पीडीवी छान आहे मला याच्यामध्ये दाखवा कोणते कोणते घेतले हा घेतला त्यांनी
Yes sir. Is it visible now? Is it okay? Okay. Hello. Someone's audio is on. Please, starting with the point where we have. Hello. हाँ sir. Okay sir, ठीक है.
यहाँ करेंगे।
हाँ मॅडम सुरू करा. स्क्रीन शेअर करा.
रहा मी शेअर करते आणि मग मला कन्फर्म करून द्याल की ठीक आहे का. त्याला मी फुल करून देऊ आहे ना, फक्त फुल स्क्रीन करते. आता ठीक आहे सर. अच्छा आता नाही दिसत आहे. तर मी तर असं करते, मी झूम मध्ये गेले, एंटायर स्क्रीन मी शेअर केली, माझ्या डेस्कटॉपचं मी केलेलं आहे. की विंडो करू सर, मी विंडो करू की एंटायर स्क्रीन करू. अच्छा सर ठीक आहे, एंटायर स्क्रीन मी शेअर केला. पीपीटी मी ओपन केलेली आहे माझी, तिला मी फुल स्क्रीन केलं सर आता. अच्छा मी तर सर फुल स्क्रीन करून टाकलं त्याला. ऍक्च्युली मी ते फुल स्क्रीन करून मग मी झूमला ऍक्सेस नाही करू शकत. हा सर. हा सर एक मिनिट, स्टॉप शेअरिंग करते परत. शेअर स्क्रीन. आता माझी स्क्रीन दिसते आपल्याला. पीपीटी आली. आता मी याला फुल स्क्रीन करून देऊ सर. आता ठीक आहे सर. ओके थँक्यू. मी सुरू करते. Yes, so all these technical glitches, and we all well aware of that with this 2020 pandemic. We all have learned that how to take care of it. So thank you again for helping to resolve that. So yes, we were talking about that we need drugs in a shorter period of time, but that drug should be very unique. It should have safety, as well as there should be no toxicity issues with that. Short-term, long-term toxic effects that has to be taken care. Like many of the drugs are withdrawn from the market. Like there is a recent example of ranitidine, which is having cardiac toxicity, or it is having carcinogenicity because of the impurities which are present in that. So these are the major issues which as a medicinal chemist or as a professional who is involved in the drug discovery process, he has to take care of all these limitations beforehand in consideration. So with this motive, we need this computational techniques, or we could say the medicinal chemistry approaches, computer drug design. So that's why this methodology is helpful to us. and that is the main purpose, that first and last purpose of this computational drug discovery technique is to have a potent, safe and effective drug in shorter period of time. Why? Because here with the help of this computer aided drug design tools, we can identify a potential compound that we can further take for synthesis, preclinical testing. If it is successful in preclinical, then we can go for pharmacokinetic and formulation development, then clinical trial. So that further journey can be started for that one drug which we have identified through the computational drug discovery method. So that's why this methodology is very important to be understood by the people who are working in the area of drug discovery. And in the last 2 years, it has taken attention of all the academic as well as research labs, so that the students can understand the concept and methodology, and they can carry forward that when they will be taking up the professional challenges. So I will be starting with very basic, is medicinal chemistry, or we can say pharmaceutical chemistry. So in this aspect, there we have to explore two things. First exploration of lead, that is identification of a lead molecule. So what is this exploration of a lead molecule? It is a chemical scaffold which is having all the plus and negative, like it is having certain biological activity, but it may be associated with certain side effects, stock parameters. There may be some pharmacokinetics or ADMET issues with that. So first our aim is to identify or to explore the lead nuclei, and that lead nuclei we can further use to further identify similar kind of drugs, and those drugs are going to have more potency and they will lead to molecule. So it will be divide of toxicity that we do in this medicinal chemistry journey or during the drug exploitation of lead moiety. So what is this exploitation? We are going to do add to any electronegative group. Do we need to have some polar chains in that, or heterocyclic nuclei? Do we need to expand the ring nucleus? So all those modifications we can do, and that can be further tested for their biological activity. So these are the basic two things, things which are there at computational drug discovery process. So next, entry करके चलेंगे. Yes, in the case of now this exploration for lead, so for that purpose, when we need to explore the lead, means we need to identify its potential for future activity. So for that purpose, this molecular modification or tailoring of existing drug, then optimization of chemical sources, synthesis of new series of compounds. So these are some existing approaches. So what we do in this case, basically, when we have to identify further new derivatives or new analogs of the identified nucleus, we can incorporate some molecular modifications. As I told you, that we can do the chain lengthening, we can add certain like if it is a or we can add different substitution groups. So that will be like tailoring of the existing drugs. We will be doing certain modification in that, like we do to the tailor and we ask him to design the fancy clothes for us. Like if there is a one particular dressing style which is in the market, we ask him to modify, or we ask him to add some certain elements in that, so that it is going to be more fancy or it is going to be more attractive. So this is also the similar approach. Here we are not tailoring the clothes, here we are going to tailor the drugs. So it is basically as per the demand. So that's why we are calling it as a tailor-made, like based on the requirement of the disease. So the drugs are being designed. So that's why lead compounds. So whatever lead nuclei we have identified. So for example, as I told you that we can lengthen the carbon chain in a certain lead molecule. But how much lengthening do we need to? Whether it is going to be methyl, it is ethyl, propyl, isopropyl, butyl, or we have how much carbon atom do we need to proceed with that? So whether it is 10, 15, 20, 2, 3, 4, so that has to be optimized. So that is one of the parameters, that whatever modifications we are going to add in the lead nucleus, so which one is going to give us optimum effect. So that also we need to decide. Then there is next approach while lead exploration is isolation of active constituents from the natural sources. Like apart from the synthetic sources, natural compound identification or drug discovery by that is, I think, well understood to all of you, because it gives us diversity in the nucleus, and those naturally available nuclei, they are having they are very specific, they are defined, as well as we like it is they are unique structures. So and they for hitting the disease targets, which are which is a very difficult task. So nature is giving us a diverse nuclei, chemical scaffolds for that purpose. Then there is next in this exploration of lead, we can synthesize some new series of compounds. So based on, like, we have optimized the lead nucleus, we have add on some molecular modifications. So we can now further move on towards synthesis of those new series of compounds, and further we can take them for like characterization and further elemental analysis, spectroscopic analysis, and then we can take them for biological studies. So this is how we can proceed with the uh, lead identification and followed by lead exploration, because it is a very important starting point when you will be starting any new project, or whenever you are going to start any work on any particular virus or any particular enzyme target for any particular disease. Now, in this process of lead identification, there are certain parameters which we keep in mind, like biological activity of the molecule. So, like, how we are going to say that the molecules which we are planning, they are biologically active. So we need to be very specific about the way which we are going to predicting that, because it is already proven that that biological activity, how it is observed, it is because of the interaction of certain bioactive substituents with the target, or with the, as I have written here, at critical reaction site of the biological system. So what is this critical reaction site? I think it is understood to you, or do I need to explain that this is critical reaction site of biological system? It means the disease targets. For example, if it is certain virus, so what kind of enzymes are there in the virus replication process, or in the throughout the viral life cycle, like polymerase, protease enzymes, replicase enzymes. So those different enzymes, that is going to be the reaction site. So biological activity is going to be because of the reaction of your molecule, the chemical moiety with the particular enzyme towards the particular ionic channel. So that is going to be the outcome of that interaction. And secondly, the biological activity results from the complex series of pharmacokinetic events. In that, we are going to consider the phase 1, phase 2 metabolism of the compounds, how it is going to be made bioavailable, whether it is lipophilic, it is hydrophilic. So, based on that, it is going to be available at certain sites in the biological system. For example, it has to be working on the CNS, so it should have good lipophilicity to cross the blood-brain barrier. So those different pharmacokinetic events, that is also an important aspect for biological activity of any compound. And being a medicinal chemist, it is our responsibility to have success at the end of the any process, that the molecule should have good biological activity. So for that purpose, we have to take care of both the things, like interaction with the receptor, and secondly the pharmacokinetic properties of the molecule, because many of the drugs, they interact very well with the biologicals, with the receptors, but because of the pharmacokinetic issues, they need to be taken away, or they have to be withdrawn from the clinical trials, from the clinical phase. So that's why these two aspects are very important. Now comes the drug action. So for the activity of any drug, like there are going to be like various parameters now, like if we are varying the structure, there is going to be change in the activity. If we are going to one single molecule, it may have various biological activities, like there are certain drugs which were first identified for one particular indication, and which were then after clinical trials were found to have effect in certain activity also. Then there is multifarious mode of action of certain compounds. They may intervene the steps between drug administration and drug response. So these are the various processes, or there are various steps in the journey of drug action. So we have to be careful with that. Excuse me. Yes. Next to that, then there is now I'm coming to the main topic, that as we have discussed about this rationale, what we are expecting from a medicinal chemist. Then we come to the main purpose. We should have a very balanced approach, and that we call as rational approach. So all our computer-aided drug design, it is based on this rational approaches. And what are these rational approaches? Like rational means which is going to be justified, which is going to be well hypothesized, which is going to be properly validated through experimental as well as theoretical methods. So we need such rational approaches, and these approaches are going to help us to understand the drug action at the molecular level. We will be able to understand the biological activity of the particular target. Secondly, it also helps us to understand the various biological aspects of various pathophysiology, how the receptor is going to be, the structure of the
बस वर्ष वगैरे बरा रिस्पॉन्स आहे. ऑनलाईन 50 जण जॉईन आहे, ऑफलाईन 50 जण झाली ऍक्च्युअली साठी आवश्यक आहे, नाही आवश्यक ट्रेनिंग
tools, X-ray crystallography, and crystal structure of the receptor. And so from there, this term has come, consider physicochemical properties. And in addition to that, we can in the chemical structure, its physicochemical properties, as well as rational approaches are very much popular. And as drug action, there are various critical reaction side. There is during the drug administration of metabolic steps, it has to undergo the drug is active while in like in case sulfate analogs that shows activity. So that drug is going to interact with the biological side, or electrolyte is going to responsible for biological activity. So that's why we say that this activity part, it is very complex, and simultaneously other regulating factors like solubility, partition coefficient, surface activity, degree of ionization, isosterism. So these are various stereochemical features of the drug. These are various properties which are going to be like, if we could understood them, we can correlate them with the biological activity, and it will help us not only to understand the this random walk or the metabolism and bioavailability journey of the drug, but as well as its elimination pathway. So that can be also understood. So then comes about like when the drug we have provided in the biological system now. So what it's going to do? Either it is going to have some affinity, or it is going to bind with the target receptor, or it is so it means we can say that it is a potent molecule, or it is going to be effective one. And secondly, it is having intrinsic activity, and it is going to show the effect. So biological activity is equal to the ligand affinity or the drug affinity with the receptors. So why now this affinity or efficacy of the drug, we can vary it based on the kind of interaction it is showing with the receptor. Like it may be acting as an agonist, it may be a competitive antagonist, it is partial agonist, it is inverse agonist. So based on the mechanism, like how it's going to interact with the receptor, we are going to predict the mechanism of action for certain molecules. So these are few basic points. Now, when we talk about the barrier, like its chemical structure, its physicochemical properties, as well as what kind of binding interactions it is going to do with the receptor or other non-target sites. So, in addition to that, there are some other parameters also, like the patient, then formulation, environmental hazards, drug-drug interaction. So, these are also other parameters which are required to be taken care. So biological active potency or dose, but also in terms of the response it has shown. So dose and response that combinedly are going to give us a measure of the biological activity of the compound. Then this rational drug divided into like we can further move towards its further you can say types. Like in that we can develop first of all some small molecules where we are they will be having some desired properties for targets, and or we can say the biomolecules. Then there may be the second method where we will be having some small molecules which will be developed and they are having predefined properties for the particular targets, their cellular functions, and there's structural information that may be known to us or we are unaware of that. So that may be the other experimental model we are having. So in the first part, that is development of small molecules with desired properties for targets, biomolecules, whose functional role in cellular process and 3D structural information are known. So that is a well developed model, and we need to we are just going to have extensive elaboration, and we could use that further. We can further modify it. So that is a very well established approach. While the second one, where the information about the receptor, about the target is not known. So in that case, or it may be known. If it is known, then the things are simplified. We know the receptor structure, we can work accordingly. But in case of unknown receptor, so there we have to be, then we need to be take some add-on features in that, and we will be using that to upscale our process. So when the knowledge of unknown targets, if it is like unavailable, so what we can do? So in the case of unknown targets, that can be obtained by analyzing the various data about the receptor which is available at the various platform, and we can use them, and we can use the advanced computation structures, gene structures. So this is about the rational drug design, where there may be available. For example, in case of we could like I am talking about drug receptor interaction. Now here we were unaware about the receptor. We were not knowing about the about the structure of the COVID virus. So how we are going to start the rational drug design? So for that purpose, for identification of the structure or the receptor structure, that is very important. So in this rational approaches, there are various approaches, like we talk about QSAR, we can talk about molecular docking, molecular simulation tools, there is virtual screening, admit prediction. So there various methods are there. So among that, the very first one, that is the QSAR, or we can say as quantitative structure activity relation. QSAR. So it is a simple approach where with the help of availability of known activity molecules, like there are what I should be saying, there are certain set of compounds which have been well synthesized, they have been characterized, and they have been put up for biological activity evaluation, and during that process they have shown certain degree of activity. So that we can say that that is our available data set, or the known activity compounds. So by using this known activity compounds, we can use the techniques of artificial intelligence, machine learning approaches, and we can derive some certain hypothesis on that, or we can identify some structural features, we can put forward some the biological activity, and that is going to give us some unknown some set of rules. And those rules which can put up on a molecule which activity is not known, and with the help of those certain set of rules which can predict it QSAR. So I think this rational lab, we are having a particular set of rules. Rules are derived from the known data, their biological activity is carried out. And that data set is high on the unknown compounds, and those compounds we will find out whether the compound is biological activity or not. So that we are. Then next, there are the new method. It has been put forward Dr. Corbin Hunch and pioneers who have they have in to apply it towards drug discovery. The models, various models. And am I audible? Am I audible, sir? Hello. Am I? Will I start? Am I audible?
Yes, ma'am.
This QSR now. So this QSR, it is a issue. Very well predicted with there are various kind of QSR mathematical modelization, discriminant analysis, PCA, nearest neighborhood analysis, then artificial neural networks. So these can be selected based on the requirement of the or the QSR which I was talking model, where we are not able to visualize certain of the developed model, like it may be in the certain predictions and visualization tools. So there was like it was quite lucrative for the chemist also to showcase the data and on the computer screen, and that was possible with the help of 3D QSAR, where we could put up the model in some beautifully placed graphical with the help of some beautiful graphical interface, the chemical structures in different configuration, different formats can be placed, and that was very easy to convey to the readers as well as to the society, that in this way we could analyze the computational outcomes, and these are going to be beneficial. So in 3D QSR, so they help us to even visualize the receptor areas, which was lacking in the case of 2D QSAR. Then comes various molecular modeling approaches. So in the modeling part, like in this case, we are having certain interface between the computer and the scientist or the person who is working on it, and we can visualize or we can rationalize the behavior of the chemical compound and its biological activity. So we could visualize that, like as I was talking about the drug receptor interactions. So that was possible to visualize with the help of molecular modeling tools. Then there are certain graphics tools, and these molecular graphics tools help to explain or to present the drug molecules and their molecular properties in a visual way. For example, you could have seen certain pictures where the molecules are shown with some blue and red areas. So, like that is a graphical presentation of some hydrophobic and hydrophilic regions of the molecule. Then there is the next terminology, that is computational chemistry, where we talk about the simulation at the atomic level or molecular properties of compounds. And those properties which will be taken care, which are going to be helpful for predicting the biological or medicinal properties of the molecule. So I think that was taken care in the first lecture. Then comes the statistical modeling that we can further carry on towards, like QSAR, that we are taking only towards the activity part, biological activity, but it is having wide application. We can use it for prediction of certain properties, as well as that can be used for prediction of toxicity parameters, as well as it is not limited only to be applied for the biologically or medicinally active compounds, that we can apply even for the agricultural compounds, pollutants, or to understand the pesticides, insecticide, their properties also we could predict based on the with the help of various QSR models. So it is having wider applicability domain. Then there is molecular data and information management systems, where all the the data which is evolved all over the world, that needs to be certain, that needs to be placed somewhere so that it can be accessed all over the world, which can help us to avoid repetition, which will help us to make the data available at one place for further processing. So for that purpose, various data banks like PubChem, Zinc data bank, then there is ligand, drug bank, protein data bank. So they are not having only the synthetic or natural compounds, but they are also providing us the data for the various biomolecules, the receptor, genes, proteins. So that has been also made available. So these various digital databases. Then in the in silico methodologies, there is one more approach that I talked in the initiation, that is about the virtual screening. So what this virtual screening is basically, so it is a combination use of the approaches I talked about, like the docking or molecular modeling. So for example, if we are having through the database, we could get a large number of compounds. So how we are going to find out which one is the active one, or which we could take for synthesis, which we could take for the wet lab experimentation. So for that purpose, we use the virtual screening platform, where we again put up certain rules, like first of all, we will collect a database. Then we will where we will have some pharmacokinetic screening for the molecules, toxicity screening. Then we will predict their binding energies. Then based on that, we will filter out certain compounds. So for that purpose, we use the virtual screening platform. Like for example, we have taken here more than potent molecules. So those can be further taken of the wet lab experimentation, and it will help in addition to these other methods also that help us to not only understand to identify and analyze the various questions which are related to the disease targets. So that help us to put up new hypotheses, that help us to apply these technologies for the newer problems. Then in silico methodologies, there is homology modeling. That is one of the approach where we could take in consideration 3D protein structure, and it help us to like work on the protein receptor moieties. So next that I would like to discuss about is about the molecular docking approach. So in the molecular docking, as it is well known that the biological activity of the drug moiety, it is because of its interaction with the disease target, and that is going to help us that is going to help us to understand whether the drug is effective or the experimentation. And this methodology we can also put up as a process in the virtual screening also, which will help us to understand whether the drug is effective or the experimentation is and for that in addition to the high throughput screening, that is one of the in vitro analysis. So similar also, which will help us to screen out a large set of methodologies. So based on this different methodology, when there might be now two cases, we may be having unknown targets, and for those based on that we could identify certain new molecules. Then there are some other approaches also, like I have depicted here. Those can be used for improve the drug-like properties of the molecule. That whether the drug is going to have good binding, whether it is going to have certain pharmacokinetic parameters. So those drug like then its TPSA, total polarizable surface area. So those can be even predicted. So that is also one of the approach or one of the pathway we can adopt. Then there are these are the certain examples of drugs which were identified based on the availability of the targets. So there are some EGFR inhibitors, erlotinib, gefitinib, imatinib. So these are certain compounds which were identified based on the target-based drug discovery. Now there are I think I am taking much time. So strategy, then there are certain strategies like if we have identified that certain moiety is going to interact with the particular receptor, but we have to like if its kinetic properties are promising and the binding is lesser, or when we have identified certain lead whose potency need to be improved, so there we can add on some ways that will help to improve the selectivity of the drug towards the target. So that can be also done. So that is a very we can say that is a trial and error approach. So in that we can take in consideration shape, electrostatic properties, flexibility, hydration. So these are the various properties which can take into consideration. Then I will like to focus on this, like for example, a drug molecule. So it may have like targeted, it may have like lesser interaction, or it may be allosteric binder. So the different kind of interaction a certain drug molecule may exhibit at different targets. So I will like to conclude here that like this computational drug discovery or the computer-aided drug design. So with the help of this various various possible methods, we can understand the drug targets, as well as we can put forward a safe and effective drug. And this approach is going to be we can apply it to develop drugs, as well as we can apply it to other areas of life sciences, like agricultural chemistry, as well as in the atmospheric, like level of pollutants in the air, in the soil, as well as in the design of the nano materials. So there are wider applications of these tools. And with this, like the basic concepts, if it is known to us, then we can efficiently put up it in our project planning, and that will help us to identify like toxic, to remove the toxic nature of the moiety, as well as to optimize it, so that those compounds can be having some tolerability during the clinical use or for the clinical studies. So thank you so much everyone for your patience listening, and I would like to have, if you have any questions, those are welcome.
Am I audible? Hello.
Hello.
Yeah. Am I audible, ma'am?
सर आप प्लीज टाइप कर देंगे.
Hello. Hello.
Yes sir.
Am I audible?
So voice, it is not continuous. It is echoing. There is some echo.
Critical mics are on here. Just a moment.
If there is any question, you can please put up in chat box. That will be nice of you.
So, any questions from the online participants? You can put in chat box. Any question from the in-person present participants? I think there are no. So no questions from the offline participants. Hello. Yeah. So thank you, Vaishali ma'am, for taking the talk. The first by Abhinav Kumar and Vaishali ma'am will definitely help us to take the hands-on training, which is planned in the post-grad session. So all the fundamental aspects related to the computational chemistry and computer-related drug design has been covered in the first session. Now there will be a hands-on training in the post-grad session. So thank you once again for taking the talk, and thank you organizer for giving me the opportunity to chair the session. Thank you. Thank you, sir. Now, on behalf of your program partners, thank you to the chairperson of this session, resource person, Madam Vaishali Patil. Madam has given us an good knowledge about the drug design. And again, I thank you all for being present here. Now, I hand over the session to Wankhede. Please.
Thank you, ma'am. for giving us such beautiful and admirable information. Now it's time to take a small lunch break. After half an hour, we will continue with this workshop. Thank you. We will meet after to this workshop after 1 hour.
क्या चीज मैम? नहीं आपका भी हो गया.
Huge difference between three. Thank you. Now what this is? Online. Thank you for this, Sarkar. So, let me tell you what we have actually planned. After the lunch, I'll come back. You do also. Okay? Will you? Some people will definitely fly away.
Hello, everyone. Okay. So after the lunch itself, do not conduct any training or any lecture session, but I do there. Okay. And after the lunch, we'll start with installing all the software required. And for your kind information, we have selected only the free and open source software. Those are free. Why only free? This is because licensed software are available. They do a good job, but you need to have a lot of money. Millions and millions of rupees or dollars you need to have in pocket. But we don't have. We are academicians. We are getting meager funding from funding agencies. We don't have amount to spend. And that's why we need to find the alternative. And the best alternative is for free and open source software system. Now, already we have shared through Google Drive and through pen drive and other media the different software required for today's workshop. There is Chem Sketch, there is OpenBabel, there is Spiral Descriptor, there is Portable Python. All these are free software, and I myself is routinely using it. That's why I am assured that with the help of this, you can go ahead. That's more than enough. So very first thing, we'll install those software. We'll understand how those work. This is because today we have chosen the 64-bit system. If you have a laptop or a machine with 32-bit system, then you cannot use them. You'll have to download the 32-bit version. Remember, all free and open software are also available in 32-bit. Free of cost. No need to pay a single penny to anyone. Not to Mohan sir also. Okay. And second thing, we'll install those. This is because those are simple Windows-based software. And in Windows, we are very good at installing the software. Just a double click will start installation of the software. Just go on next, okay, and ultimately it will be installed. So this is the simple procedure. Because these software are used, actually are now still used, and I was eager to share those. We use Google Drive. That's why there are two versions you will see. One is the zip version, the another one is open version. Okay, because internet is full of different types of motor bacteria and viruses. That's why we have to be very careful while sharing. That's why zip version is also there. You can extract it with the help of WinRAR or WinZip. Both are needed to extract or unzip them, and you can use them in your PC. Remember, earlier in the morning, we got a request from Professor Dr. My very good friend of mine, Professor Dr. What's the name? Somebody asked about Android system. Anyone? Somebody asked about Android system. So remember, what about Android system is that your Android phone is very, very good. It's about single processor based. It may be octa-core or hexa-core, but it's not useful for doing this type of stuff. Because if you start this, okay, then it will melt down. Remember, it consumes a lot of energy, a lot of heat is generated. That's why you need to have PC with you. This is reality. Okay, you can do this is because Android itself is a type of Linux, and all softwares are available free of cost in Linux system. We can use it, but that's going to be more a command line version than the actual graphical user interface. So if you have GUI, it's very easy to use them. So in our real training session, we'll use Windows, whether it is 7, 8, 10 or 11, it should be 64-bit. We'll install them, and then one by one we'll go ahead. The very first thing is about the student fraternity here, because they want to know, because this is the beginning. We'll start with Chem Sketch to draw the different structures. We'll use the MM, molecular mechanics, to optimize the geometry, how to save them in different file formats. Those are very important. This is because you want to communicate with another scientist. If you want to share your data, say for example, a person from Europe or US, then you have to communicate it at a proper platform. You have to share your data in the form of structure, and appropriate file format is required. So you'll understand what type of file formats are available, what is the basic structure, how to save them and use for communication. Then we'll go for the next part, use of different types of file formats to calculate admit properties using different easily available, free of cost web servers. Everything is going to be free. Nothing is costly. Okay. Then the next part will be to use this to generate the pharmacophore model. Those are the important aspect of computer-aided drug designing. And then we'll go for actual QSR. And if time permits, we'll touch the big arena of molecular docking. That we have actually planned, but planning never executes successfully. But for this successful organization and conduct, we need to have your presence with only one thing, that is silence, and it's very costly. Okay, remember, silence is the best thing you'll have to maintain throughout the training session. If you have any query, then you can ask very good friend of mine, Dr. Vijay. He will be with you throughout this. I might be busy with my laptop and engaging the participants from other states and countries, but he is with you. And I'm not good at doing all these things. So what I will do, wherever I'm more stronger, I will hand over the mic to my friend, and then I will be with you. Okay. So this is what I actually planned. So throughout the session, we'll be wandering here and there, one-on-one interaction. If you have any query, you can ask. We'll show it on the screen. We'll try to solve it. But before this is my humble request to try to install all the software of your own. You will learn how to install them. And one more thing, if you have your own laptop, you can use those at your home. So whenever you are finding sometime, you should take screenshot. You should note down something. And the best thing is to read the manual of each and every software. We all do this in our laboratory. Before starting any practical, we do read the manual of an instrument. This is called as genuine good practice. We'll follow all these good practices here, so that at the end of the day, you will be able to have something in your pocket which you can then use in your research, have quality output, some good content, and then it will be useful for you to have quality publication in either UGC Careless Journal or Scopus based or SCI journal. We are aiming at quality output. For this, we have planned meticulously. Your support, enthusiasm, and your queries will make this to be useful for each and everyone. Okay. So this is all about what we have planned, what we are actually thinking. And if you have any query, then we are here. And even after this event, you are open to ask any type of question to me. One thing I forgot. That's very simple. Earlier in the morning, we listen two different lectures on different arenas. One is computational chemistry, very core, touching the quantum chemistry aspect, requiring a lot of computational usage, heavy machinery of high RAM, maybe 32 or 64 GB RAM. Okay, high level of processors are required, but that is not required in computer-aided drug designing. That was the part covered by Dr. Vaishali Patil. I'll continue this, and now on your laptop you'll actually see that nothing is required to do this. So don't worry, whatever you listen in the morning, that is to create a platform, increase your understanding, and so that whenever you will execute the software, you will understand why this is actually happening. Without the basic knowledge, you cannot go ahead. When you write the paper or put your results in front of someone, you need to have some understanding. That's why these two lectures were there. They have touched, they have covered broad area in small time. And one more thing, in a single day, we cannot make you an expert. Rome was not built in a day. Do you agree? So in one day, We cannot make you expert, but at least we can give you something that will be with you forever. I hope that is what the intention was in your mind when you registered for this workshop and you are here. Am I correct? No. Yes, that is the spirit. And we say you should share your queries. Okay, very politely, very silently, so that we will be there. And I'll try to have some arrangement that by wandering here and there, I'll be with you, interacting directly and solving your issue. Okay. So please do the testing. So with this, the planning is with you, and after the lunch, we'll redo join this, and before this, at least we'll inform each and every person to join online as well as offline, so that in this hybrid mode, we'll be able to successfully conduct this workshop. Any question from your side? Query? Did you get anything from my side? It's just a planning. Okay. So after this, yeah, one more thing. With the kind permission of the organizer, I'll start with one of my own lecture. Will it be fine? So that you can understand what actually we are doing and why. Is it fine or not? So we'll join after the lunch with a nice lecture on computer-aided drug designing, focus on two technologies, QSAR and molecular docking, and a little bit of pharmacophore modeling. Okay, so I hope you understood what we are actually going for, what we have to do. That's all. After the बंद करा झालं सर हे कसं करता येईल त्याला कनेक्ट नाही ते तर झालं बंद करा सध्या